The overall goal of this experiment is to digest fluorescently labeled DNA molecules with methylation and non-methylation sensitive restriction enzymes and determine the digestion rate of the experiment.
This method can help answer key questions in the chemistry or biology fields, such as genomics.
The advantage of this method is that a scientist can digest pre-stained DNA with an enzyme to determine if the DNA is full-length after the completion of a different experiment. Demonstrating the procedure will be Drew Thompson, an undergraduate student from my laboratory.
To make a 0.7% agarose gel, weigh out 1.4 grams of high gelling temperature agarose in an Erlenmeyer flask. Then add 200 milliliters of 1x TAE buffer and place an inverted beaker on top of the flask. Heat the solution on a hot plate until bubbles begin to form on the bottom of the flask and float to the top. Remove the solution from the hot plate and let it cool until the flask can be touched with a bare hand. Then pour the gel into a 24.5 cm by 21.8 cm gel mold with a gel comb to create wells. Use a pipette tip to pop the bubbles near the comb or on the surface of the gel and allow the gel to solidify for at least 15 minutes. To stain lambda DNA at 6 different concentrations of dyes, begin by using 1xTE to prepare 300 microliters of 100 nanogram per microliter lambda DNA. Store the solution at 4 degrees Celsius. While preparing a sample for each of the following dye concentrations for